now. Okay, so today just going to continue on talking about event-related potentials. Um, First, I do want to say, just remind you about Group Project 2, that you do need to be thinking about coding your experiments inside Toolkit. I know some groups have already started. One told me that they basically are done, and that group I believe I'm meeting with after class today to actually look at their code, and that should be cool. So please remember that's definitely something that you need to be working on and hopefully completing here in the near future is coding your study. Remind you also that you'll have to collect data. Most groups will be collecting data from at least 30 participants. There were one or two groups that have a between subjects factor, and I told those groups that they could collect data from 40 participants, and that would be sufficient. So that's a little bit of a variation on what I talked about previously. I'd originally said some groups might, if they have a between subjects factor, would have to collect data from 60 participants, but I'm making exceptions for based on the conversations I have with those groups where they will only have to collect data from 40 participants. And I also should mention, just like with Group Project 1, you will be turning in your data files for your experiment for Group Project 2. One group, I know we kind of decided they might be manually scoring their data instead of using the SciToolkit data files. Um, but for that group, I would like to see, your manual scores of your participants, the actual things that you wrote down, if you're doing it that way instead of scoring things inside Toolkit. But either way, I definitely want to see all of the data files for Group Project 2 when you turn them in. I should also mention that I have graded your Group Project 1 second half papers. And I have provided feedback, pretty detailed feedback. Usually each one of these takes me like an hour to grade, so I definitely read it closely and provide feedback. So I've provided feedback in the document, or except for the one group that gave me a PDF, I just wrote it in the email. And then I sent an email to the first author of each paper. So please, that first author, forward it to the rest of your group. If the first author on the paper has not forwarded it to you, and you would like to see the paper, the grade, and the feedback, and your other person hasn't given it to you, simply email me. I will be happy to forward it myself. General, the papers were pretty good. There were still a couple groups that didn't seem to have their head around exactly what a hypothesis was. For Group Project 1, remember we did hypothesize the type of sound by congruency interaction. There was clearly some groups that even after the second half paper still didn't really seem to have their head as to exactly what that interaction would have meant, exactly what our hypothesis was. But most of the groups did seem to have a little bit handle on that second time around. Probably the biggest issue, I think almost every single group had this, was in the method section. And because almost every group did this, I did not take off for it. But the biggest issue that I probably saw that almost every group had was that almost every group did not mention the specific task that participants are at. There might have been one or two groups that did mention this. In the procedure section, you need to make sure that you mention the specific task that participants perform on each trial of the experiment. And the Stroop task, the thing that participants are asked to do is to indicate the color of the text or letters that compose the word on each trial. And most groups did not mention that. And in fact, in general, I would say, while you guys did a very good job talking about the stimuli regarding the Mozart and rain chime stimuli, for the most part, everyone kind of talked about those and said half the participants heard one sound, half the participants did the other. There was very little description of the Stroop task. I kind of get the impression that the reason, perhaps because I did most of the coding for the Stroop task, you kind of forgot about it when you were talking about your procedure section. But that was when you guys are doing your Group Projects 2 papers, definitely encourage you to be a little bit more detailed in explaining exactly what task the participant is performing, responding, indicating the color of the text or letters compose each word by pressing 1 or 4 buttons on the keyboard. Yeah, that's really it. That's kind of the general feedback. If you have specific questions about the feedback, I'd definitely be happy to meet. That's definitely a good use of the time. I give you feedback for this paper because I'm hoping that it will help you for group project 2. And I do appear, it does, some of these papers were way better on the second draft, on the second half than the first draft. So I do get the impression that the feedback works. Some of the groups, though, appear to pay more attention to the feedback, and others not all groups made a lot of progress from the first half to the second half. But I definitely suggest thinking about the feedback that I've given you on these first 2 papers that you've written and incorporating it for group project 2. Oh, and one other thing, there are still some groups that had some formatting issues. And so those are one of the most easy ways to take off points, because there's an objective right and wrong there. So please make sure you focus on formatting for group project 2. And if you have questions about formatting, I would be happy. That's a very easy thing for you to make a quick appointment and be like, hey, look at our formatting, see if it's okay. And if it's okay, I can look through it in like 30 seconds, and I can get an idea if the formatting's okay or not. So I'm happy to help with those types of things for group project 2. Okay, so now I'm going to talk about event-related potentials today. Oh, and I guess I should also mention, there's the quizzes. I mentioned this in an email last week, that you still have quizzes, and basically I will always be sending emails when I posted the quizzes on Blackboard, and I will give you the due date. And all of the quizzes at this point are going to be due several days after they are listed in the syllabus. Basically, we've gotten a little bit behind, especially got behind that week when I was sick, keeping up with the quizzes. So I will be sending an email today. In fact, later today after class, I will finish those a couple quizzes, and I will post them, and they will all be due towards the end of the week. Maybe even, I think I'm thinking leaning towards like Friday or Saturday for those quizzes. Okay, so now we will talk about event-related potentials. So far, we have talked primarily about brainstem potentials with respect to hearing sounds. And remember, the brainstem potentials have a really, really short latency. Let's see if I have a graph here. Yeah, so here's an example of a brainstem potential. This V and A, these are supposed to be kind of the first components of this brainstem potential. The stuff before is like the baseline period when nothing's happening. But I want to point out that there's definitely a tiny bit of a delay. A sound occurs at time point 0, but then there is a delay. It's sometimes estimated to be as small as like 2 to 3 milliseconds in the brainstem potential. In this study, it looks to be at around 7 milliseconds, but that's a really short delay. 10 milliseconds is a 1/100th of a second. And so the delay from the time a sound starts, the onset of a sound, until the time that your brainstem starts encoding information about that sound is less than 100th of a second. But there is a little delay. There is a tiny bit of a latency. And this is one of the main things about brainstem potentials, is that they have really short latency. And that's kind of how we're going to be going. We're going to be going through all of these different event-related potentials that are shown here. And kind of the main thing, especially about these ones in the tightly time-locked group, which is where we're starting with, is that we're going to start with the really quick latencies, like the short latency event-related potentials, like the brainstem potentials. These have short latencies of a sliver of a second. And then basically we're going to be moving from later and later to we get to these P600, which we're probably not going to get to the 400 or 600 today. But basically what we're doing is we're just kind of moving further and further into the latency. So starting with the really quick latencies, brainstem potentials, the sensory ERPs today, they have a relatively longer latency, somewhere between 60 to 100 milliseconds. The mismatch negativity, another event-related potential, has a latency of like 150 to 250 milliseconds. Also going to be talking about the early right anterior negativity, another event-related potential. You read an article about all of these, sensory ERPs, P1, N1, the mismatch negativity, you read an article about. The ERAN, early right anterior negativity, you read an article about this. These have a latency between like 150, 200 milliseconds, maybe 150 and 250 milliseconds. The longer latency ERPs, these are really easy to know their latencies because, it's part of their name. P300 is a positivity with a latency of 300 milliseconds, N400 negativity 400 milliseconds, P600 a positivity with a latency of 600 milliseconds. So you can see we're moving from really short latencies to relatively longer, and that's kind of how we'll go through all these first ERPs that I'll be talking about in this portion of the course. So once again, these are the same slides that I was using last time. I just keep on adding to them, and eventually there'll be one long slideshow, which is basically the entire event-related potentials unit all in one slideshow. So that's where we left off last time with that extra credit thing that I talked about. And so this is where we're going to begin today. And you did read an article about this. The event ERP Review 1 was an article that was about these kind of sensory event-related potentials. And we'll be getting into the longer, P3 would be considered kind of getting on the longer side. Obviously P1, N1, P2, these are the sensory ERPs. And then as the latency gets longer, we start getting into the longer latency event-related potentials. This kind of naming scheme, which is common: P, positivity, N, negativity. P1 is the first positive peak, N1 the first negative peak, P2 is the second positive peak. So remember, sometimes you'll just see like a single number there in the name of event-related potential, which means it is the first, second, or third bump, or the first, second, or third peak in the waveform. And in some event-related potentials, instead of just having a single number, they'll have like three numbers there, like the P300, which is a positivity with a latency of around 300 milliseconds. So sometimes these numbers will represent like the first, second, or third peak. They're counting the amount of peaks. Sometimes these numbers here, though, represent the latency in milliseconds of the peak. So they're kind of different naming schemes for event-related potentials depending on basically which field you're in. So now we'll move more to talking specifically about those sensory ERPs. So this is a screenshot that is taken from the ERP Review 1 article. So this is one you guys took a quiz on it last week, I believe. And it's showing two waveforms that are kind of on top of each other. And what the study here is looking at is how do these sensory event-related potentials change in response to a stimulus, depending on when that stimulus is presented. Are you paying attention to it, or are you paying attention to something else? So this is a pretty basic question in cognitive neuroscience. How does paying attention to something influence the way the brain processes that thing? So the thing which is being processed in this study is this vertical black bar right here. The participant looks at the fixation cross in the center of the screen. And then on each trial, a black bar can actually occur in the left visual field, or a black bar can occur in the right visual field. These data, though, right here, these are the neural responses to when the black bar is presented in the left visual field. And it happens to be that this is an idealized waveform, presumably taken from a right hemisphere electrode. So the stimulus was presented in the left side of the world, and this electrode says idealized. So this is basically what the researchers see in most studies when measuring responses in the right hemisphere. Usually it'd be like the right occipital or right occipital parietal area. The key factor, the key independent variable in this study, though, is you can see here the arrows are kind of pointing. Attend left. Those are the trials. The vertical bar occurs right here. And when it occurs, the participant is attending to this exact point in space. Compared to attend right, the vertical bar still occurs here in the left visual field. But when that bar occurs there, the participant is paying attention over here. And then the researchers are looking, how does the neural response to this bar change depending on whether the participant is paying attention to right here when the bar occurs, or if they're paying attention to right here when the bar occurs. And these findings are pretty straightforward. I don't think they're particularly complicated. The neural responses appear to be enhanced when the participant is paying attention to the point in space that the vertical bar appears. Attend left, that's the solid trace right here. We're looking at the P1, the first positive peak. And the amplitude for the P1 is bigger when the participant is attending to the left, when they're attending to the bar, compared to when the attend right is the dotted line, which is right here. And the P1 amplitude is relatively smaller when they're not attending to the stimulus when it occurs. And you can kind of see those differences persist to the N1 as well. So the N1 is negativity, the first negativity peak. And you can see there's a difference there. And so you can see for the P2, there's also a difference. And in each one of these, basically the waveform, the amplitude is bigger, either in the positive or negative direction in response to the stimulus when the participant is paying attention to it. So it's a pretty straightforward finding. So there are some intricacies, though, of this review article, ERP Review 1, that you read, and they're pretty important for understanding and appreciating event-related potential studies. So let's see, we'll talk about several things here. First, I want to show you this brain. This is not a perfect brain picture. I found it on Google. I mean, it's accurate, and it's pretty good. It has more information on it than I kind of wish there was a simplified one, but this is the best one that I could find. I have a question here in the chat. Let me take a look at this. Are there studies with event-related potential while participants perform the Stroop task? I would almost guarantee that there are. I would almost guarantee that there are, but I have not happened to have read any of those. Good question. I don't know that. I've never read any of them, but I'm sure there's probably like 50 to 100 of those, I would imagine. Okay, so the brain, we're talking about here for this event-related potential study, the review article that you read. Mostly the brain regions that we're talking about are back here. And so this is the occipital lobe back here. And please remember that the occipital lobe, the sense out of the 5 senses that it's most kind of connected to is the visual modality, the sense of seeing. Primary visual cortex is in this area, and primary visual cortex is also called V1, which is also known as striate cortex. Striate, by the way, refers to the visual appearance. So if you were actually to look at this part of the brain, you open up a skull and are actually looking at V1, that is primary visual cortex, it appears to have like a striated, which I believe means striped appearance. And that's right here. One thing I want to point out about V1 is it's located medially, which means towards the center of the brain. This right here, the longitudinal fissure, divides the brain into the left and right hemispheres. V1 straddles that fissure right up to right up there, right in the middlemost part of the brain. That's primary visual cortex right there, kind of the most medial part or center part of the occipital lobe. It's also very low in the occipital lobe. That's striate cortex, V1, primary visual cortex. It basically has 3 different names. And I'm bringing all this up because the ERP Review 1 article that you talked about, it makes the distinction between stuff that's going on in primary visual cortex, V1, striate cortex, and stuff which is going on in kind of the surrounding regions. And so those surrounding regions are shown here like V2. That's V2 right there. V3 is the blue one right there. V4 is right here. V8, I actually don't know much about V8. And then V5, yeah, right here. So there's various all those are different visual regions. V1 is the primary visual region. V1 is striate. All of these other visual regions, though, are called extrastriate, meaning they don't have that striped appearance. Only V1, primary visual cortex, is striate. All of the others are extrastriate. I guess that extra means outside of. And the ERP1 review article kind of makes the claim that the extrastriate areas are basically, you can kind of consider them relatively more lateral than V1, that is striate cortex, which is medial. And this makes some sense because there's a lot of theories which basically propose that light enters your eye and then it does some things in the brainstem. But then eventually the first cortical area that visual information goes is V1, which is right there. Goes from your eye and then to V1. And then from V1, that information is kind of thought to travel out in the sides of both hemispheres, left and right. It's traveling from that medial place of each hemisphere, V1, and then it's kind of traveling in a lateral sense to the temporal lobe, which is right here. Here's the temporal lobe. So you can imagine the visual information starts here, and in the left hemisphere it goes out this direction, and in the right hemisphere it starts here, and then is wrapping around in the right hemisphere, starting in the occipital lobe, V1, and then going to the extrastriate areas, and then eventually reaching the temporal lobe where further types of visual processing occurs. So this is kind of the anatomical basis for a lot of the stuff that was being described in this ERP Review 1 study. So they were talking in this study about the P1 latency. So the P1 is something that occurs, we just showed you right here. The P1, the first positive peak, that occurs in response to a visual stimulus. So keep in mind, when we're talking about the brainstem potentials, those are usually measured with respect to a sound. Now here we're talking about the P1 ERP, which is most often studied with respect to visual stimuli. And so they're studying, and they were talking about in this review article that you read about a bunch of different research about the P1 event-related potential. So a couple things about it. The latency is around 60 to 100 milliseconds. So one first thing to point out is that latency is longer than the brainstem potentials. The brainstem potentials is like 2 to 10 milliseconds, really quick. The P1 latency is relatively longer, 60 to 100 milliseconds. So we're moving longer and longer latencies. That's kind of the path we're taking in this unit. As you just saw in the previous article, P1 amplitude is modulated by attention. And a fundamental question is, what are the regions of brain where attention does stuff? You know, attention clearly helps us process things out in the world. And clearly the brain is involved to some extent. But what regions of the brain are actually influenced by whether you are paying attention to something or not? Is it V1, that is striate cortex, or is it more of the lateral, the areas outside of V1, extra striate cortex? So they know that the P1 latency is, the P1 amplitude is modulated by attention, but what exactly are the brain regions that the P1 reflects? What are the sources of this P1? And remember what we've been talking about, that the EEG methodology, that is the whole idea of measuring electrical activity on the scalp, is it's difficult to localize where in the brain that electrical activity is coming from. So just because you see electrical activity, you see electrical activity in the scalp, you don't know where the source is in the brain. However, the article that you read, this review article, they made the claim that the P1, they believe it's coming from extrastriate cortex. Extrastriate cortex. And so you should be thinking, why, if we can't localize the source of electrical activity, how do they know it's coming from extrastriate cortex? So I'll get to that in a second. The C1 ERP is not modulated by attention. So this is, P1 amplitude is modulated by attention, and it's thought to come from the extrastriate cortex. C1 is another ERP, which they talk about a little bit in this article, but it's not modulated by attention. At least according to the article, they said they have data indicating the C1 event-related potential. It processes a visual stimulus the same, whether you are paying attention to that stimulus or not. And they said that they believe the C1 event-related potential is coming from striate cortex, that is V1, primary visual cortex. So the question is, which is a good question, if EEG is so bad at localizing the electrical signals to specific regions of the brain, why do they believe these things, that the P1 is coming from extrastriate cortex and the C1 is coming from primary visual cortex? How can we know where these things are coming from if we can't localize them to specific parts of the brain? And the evidence is, I guess you could say, circumstantial, or it's not perfect evidence, but it's pretty compelling and it's interesting to think about. So here's a couple pieces. They talk about, in box number 3 of the article, they go into great detail about the evidence regarding the sources of these P1 and C1 event-related potentials. So the first thing, the C1 event-related potential is thought to have an earlier latency. Unfortunately, they did not show a picture of this event-related potential in the article, so I don't have a picture to show you either. But basically, they say the C1 event-related potential has an earlier latency. And remember, the order in which I said visual information is thought to go through these regions. It first hits V1, and then it's thought to travel out to these extrastriate areas, especially towards the lateral, the sides of the brain. It starts in the middle of the occipital lobe and then moves out into the lateral or to the sides. And the C1 ERP has a relatively earlier latency of 60 to 80 than the P1 ERP. And remember what I said, most studies indicate that the P1 ERP has a latency of like 60 to 100 milliseconds. So 60 to 80, basically most studies are showing the C1 ERP is just a little bit earlier of a latency than the P1 ERP, which is just a little bit later on average. And so this suggests then that the C1 ERP, it's probably coming from an earlier visual region like primary visual cortex, whereas the P1, that's probably occurring from a relatively later visual region such as V2 or V4 or V5. That is those extra striate regions. C1 is coming from striate region, V1 presumably, and then P1 is thought to occur in the extra striate areas, the ones that are on the sides of the brain more. And then this is probably the best evidence that they talk about. This is the evidence that's probably the strongest, because there's this thing about V1, primary visual cortex, that is striate cortex, which appears to be unique. And then they said such a good job. It was difficult to paraphrase because they just said it so perfectly. I won't try to read it. I will try and summarize the most important thing. So this topographic mapping of V1, this is the idea that there's an upper visual field and a lower visual field, and that those are represented in V1, primary visual cortex. But there's some evidence, presumably it's some evidence that's not based on EEG, that the upper and lower visual fields, and I do want to quote this part, are oriented 180 degrees from each other in the calcarine sulcus. So I plan on showing you anatomically where this is. What you just need to understand basically is that in the visual in V1, the upper and lower visual fields, what's up here and what's down here, they're kind of anatomically separated in primary visual cortex. And that makes an experimental prediction. And the prediction is, ERP activity that's generated in V1 should be opposite in polarity, meaning positive or negative, for upper versus lower visual field stimuli. So for instance, if upper visual field stimuli tend to have a positivity, then if you present something in the lower visual field, it's going to tend to have a negativity. If that occurs, that indicates that you're probably measuring signals from V1. V1, striate cortex, primary visual cortex, seems to be unique, where when visual stimuli go from the midline, above the midline to down below the midline, so midline would be like right here, above versus below the midline, the signal, at least in V1, changes from a positivity to a negativity. V1 seems to be unique in this. And so consistent with this prediction, they find that the C1 wave is, so I said it backwards, I said positive up here and negative down here. I got it backwards. It's negative for the upper visual field and positive for the lower visual field stimuli. So the fact that the C1 wave has this property, stimuli up top tend to have a negative polarity, stimulus down at the bottom tend to have a positive polarity. The researchers believe that indicates that this C1 event-related potential is probably coming from primary visual cortex, V1, striate cortex. So this finding suggests, C1 coming from V1. C1 is coming from primary visual cortex, while the P1 ERP is thought to be coming from a relatively later visual area, particularly those extra striate regions. So I guess that's all I have to talk about regarding. So this is the evidence that the C1 comes from primary visual cortex, and P1 is thought to come from the extra striate regions. So now we're moving to another event-related potential, the mismatch negativity. The mismatch negativity, so this one has a relatively longer latency. As I said, we're starting with the shortest latency event-related potentials, and we're just moving relatively longer and longer event-related potentials. And the mismatch negativity is pretty darn famous. When you read an article, you read a mismatch negativity review 1 article. It's bolded right there. So as the name implies, mismatch negativity, the mismatch MMN is a negativity, and it occurs in response to a deviant stimulus. That is it. Sometimes it's called a deviant stimulus. Sometimes it's called an oddball stimulus. And it's usually measured with sounds, though I do believe you can measure one of these things with visual oddballs. It's usually auditory oddballs. So the types of studies that I've read the most of these, when you're just presented a pitch over and over and over again, like me, 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 that high-pitched one that I did, that would be considered the deviant. So you hear the same stimulus over and over and over again, a pitch of a certain sound. That's the standard. And then eventually there'll be another pitch, which is, higher in pitch or lower than pitch. It's just different than standard in some way. And then you compare, how does the brain's response to that deviant stimulus, how does that compare to the brain's response to the standard stimulus. And that's what the mismatch negativity is. It's measured by comparing the amplitude of the responses in this window, usually between 100 and 250 milliseconds after the onset of each sound. As I said, it's often a pitch. Sometimes it's speech stimuli, so it might be like da 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 ba da 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 ba da da da. So that would be an example with speech. It's either pitches or speech, or it can be other things. It's usually measured in a latency of 100 to 250 milliseconds after the onset of the stimuli. And it's the negativity is bigger in response to the deviant stimulus. So the response to the standard is pretty small, but the response to the deviant stimulus, there's a big or relatively bigger negativity that occurs for the deviant stimulus compared to the standard stimulus. So the article that you read, it kind of suggests kind of two different things about where this is located as far as the electrical activity on the scalp. And they say frontal temporal at one point in the article, and then they also say frontal central in a different part of the article. So these aren't necessarily, these are not mutually exclusive by any means, but they're kind of a little bit, and they say two different things, but they're kind of a little bit of overlap. Frontotemporal means frontal and temporal. Frontocentral means frontal, but more towards the top part. And both of these are kind of true. If you actually look, and I'll show you some scalp maps here in a second, you kind of find the mismatch negativity response and kind of like the front, and like 60% of the brain. Really the only place that you don't find the mismatch negativity is like way here back in the occipital lobe. You don't find much. And I guess in the inferior parts of the temporal lobe, you don't find much. But in like the superior parts of the temporal lobe electrodes, and in the top part of the brain, and then definitely in the frontal lobe, you see the mismatch negativity response. So I keep saying frontal lobe and temporal lobe. I'm not trying to imply that these ERPs are definitely coming from the frontal lobe or the temporal lobe. I am simply talking about the electrodes where these responses are strongest, and the temporal electrodes, especially in the superior side, the frontal electrodes, and then the central electrodes. Those are where the response is most strong. So frontotemporal and frontocentral electrodes. So a couple little facts about this, and I'll go into detail about some of them. The mismatch negativity amplitude is smaller in schizophrenia. So one of the things that, researchers are very interested in is, there's a lot of grant money from the government trying to find biomarkers, biological activity, which can help to diagnose different conditions. And the mismatch negativity amplitude is pretty consistently reduced or attenuated, weakened in schizophrenia. And that was one of the things. I think I had a quiz question about this article. And basically, there's been they talked about in the mismatch negativity review 1 article, there's like 30 different published studies which show that the mismatch negativity amplitude is reduced in schizophrenia people compared to controls. So that's an interesting fact. And I'll show you some evidence in support of that here in a second. I'll show you one of the studies, in other words. And then another thing which is really important about the mismatch negativity is probably one of its most important theoretical characteristics is that it is pre-attentive. So pre-attentive means that it can be elicited even if you're not paying attention to the sounds. So remember the mismatch negativity, larger negativity that occurs in response to a deviant sound compared to a standard. And that larger negativity occurs even if you're not paying attention to the sounds at all. And so the way they know that is because very common in the mismatch negativity studies is the participants will be paying attention to something completely other than the sounds. Usually it's a visual thing. So they'll have the participants watch a silent movie. And then participants will be wearing headphones, and that's where the beep 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 beep. That's where that's occurring in the headphones while they're paying it. That's really boring, really repetitive. And so they'll give the participants a silent movie to kind of watch. Presumably they're kind of interested in that. And even when the people are distracted by watching a silent movie, you can still find these differences in responses to the deviant and standard stimuli. That's what is meant by pre-attentive. You don't have to be consciously processing the sounds in order for the brain to respond differently to the standard and deviant sounds. So here's kind of the way, this is kind of the scalp topography. So each one of these little things are kind of hard to see. So, you might want to take these slides some other time and you can actually make the picture bigger if you want to get a closer view of it. But each one of these little things right here, those are electrodes. So this is a group of researchers who did the study that have, like 100 different electrodes, so electrodes like every other quarter of an inch or so on the person's scalp. So lots and lots of electrodes. And then you could see, if you look closely, and they kind of zoom in for one of them right here, there's a red trace and a blue trace. And you got to look at the legend here. The blue trace is the ERP standards, and the red trace is the ERP deviance. And then consistent with the latency, so this right here is basically the peak amplitude of the mismatch negativity response right here. So in this, keep in mind, this is pretty consistent with what I mentioned. The latency of this is usually around 100, 150 to 250 milliseconds. And you can see though that the kind of the difference between the red and the blue traces, it kind of lasts throughout this whole thing. Here, it looks like it kind of ends at around 200 milliseconds. So from around, in this particular study, from around 100 to 200 milliseconds, they're showing the difference between the stimuli, and the direction of the difference, the polarity of the difference matters. The ERP deviants are relatively more negative. The red are more negative than the blue. If you notice, the down is negative, up is positive. The red is more down. That is, the ERP deviants are more negative than the ERP standards, the blue. So more negative. That the difference between these two things right here, that is the mismatched negativity, those differences. And though this is what's shown here is really important for understanding how to read graphs in ERP studies. These kind of graphs are pretty straightforward to read because you can see the two different conditions, red and blue, deviants and standards. However, a lot of ERP studies will show these graphs. And what this graph represents is a kind of running difference score between the red. So notice how there's two things here, but in this one, there's just a single thing. And what this thing represents is you take for each individual point in time, and there's, you're breaking up this 400 milliseconds into small, incredibly small little slivers of time. For each individual sliver of time, they're taking the amplitude of the deviant minus the amplitude of the standard, and then that's what they're plotting right here. And so like the mismatch negativity is basically this, this part about right here. This is where there's a difference right here. And you can see now the negative is up and the positive is down. So basically parts in this where it's non-zero and it's in the negative direction. This is basically the mismatch negativity, but this is a difference wave. It's basically taking the deviant amplitude minus the standard amplitude. You get some number. You do that for each time point between 0 and 400 milliseconds, and you plot it here, and that's the difference score. This is a common way of kind of having to read graphs. The single line represents the difference in amplitude between these two lines right here. So the difference is kind of in a running total right here. Let's see, moving on. Yeah, so here's the study with schizophrenia. So this is why, as I said, there's many, many studies which support this. This is just one of them. I also like to show this one because it once again shows all these different electrodes. This group, these researchers don't have quite as many electrodes as the previous study, but they still have a lot. And then, it's a kind of a similar overall structure of these experiments. This is one where, control participants versus individuals with schizophrenia. And you see the two different traces. Each one of these traces, though, is a difference wave. That is, each one of these is kind of like, this thing right here. But in this graph, they're showing a difference wave for the controls and a difference wave for the patients. And you can see the evidence of the mismatch negativity because if you look at the controls, they're the dark solid line right here, and the patients are the relatively lighter line right here. You can see that the controls responses are relatively more negative than, the controls responses are relatively more negative than the patients, that is the individual with schizophrenia. And you can see that actually from most parts of the brain. Please notice though, that when you get to like the occipital and parietal, some of these lower parietal electrodes, the response kind of becomes weakened or attenuated. I guess the exception is kind of like right here. It's still kind of strong. But if you look, generally the differences between the light bar or the light line and the dark line, really, really strong at those frontal, central, and some of the temporal positions. It does get a little bit weaker when we get more towards the back of the brain, which is consistent with what was said in the Mismatch Negativity Review 1 article. It's a frontotemporal / frontocentral electrodes is where this is strongest. And that appears to be somewhat represented here in this figure. But you can see this is probably the easiest one to read right here. There's, if these are difficult, you can basically look, this is the mismatch negativity amplitude. Remember, that's the difference in amplitude, deviant stimulus minus the standard stimulus. And you can see for the controls, a very negative number. The patients, people with schizophrenia, it's a negative number, but it's not quite as, it's a smaller negative number. This is very negative. This is relatively less negative. And that's consistent for all of these, each one of these things, PZ, CZ, FC, FPZ. These are just different electrodes, and they sample different electrodes on the brain. And basically wherever you're measuring, controls had a bigger mismatch negativity. That is, their response to the deviant sounds was more negative to the standards. And that difference was stronger in the controls than it was for the individuals with schizophrenia. So this is, the mismatch negativity is a very common biomarker for schizophrenia. If an individual has schizophrenia, they likely will have reduced amplitude of their mismatch negativity. The difference in amplitude between the deviant and the standard stimulus tends to be smaller in individuals with schizophrenia than controls. Okay, so I'm talking a little bit more about, I got about probably 10 or 15 minutes more of stuff. And I wanted to talk a little bit about this theory, which is talked about in the Mismatch Negativity Review 1 article that you read for last week. And particularly, they talk about this adaptation hypothesis. So the purpose of this hypothesis is kind of trying to explain what causes the brain to respond differently to the deviant and standard stimulus in the mismatch negativity response. The brain's clearly, the electrical activity of the brain clearly can differentiate between the deviant and standard stimulus, where for at least controls, their response of the deviant stimulus is more negative than it is to the standard. In other words, for controls, there's more brain activity in response to the deviant than there is the standard stimulus. Why exactly is that? And so there's kind of two hypotheses which are really focused about in this review. The one which has got, that they talk about, which they don't agree with, but this is a common hypothesis, the adaptation hypothesis. So a common thing in the brain is that the first time you play a stimulus, you present a stimulus, the brain responds very strongly to novelty. But then if you keep playing the same thing over and over and over again, which is what happens to the standard, the brain adapts. Or in other words, the brain's response to that stimulus decreases the more it hears it. It's almost as if the brain's getting bored with hearing the same thing over and again. The first time the brain is interested in hearing something because it's new, it's interesting. But then when you keep hearing the same thing presented over and over and over again, the brain basically loses interest, and you can see that by the fact that the brain's response is reduced. The brain's response to the standard is reduced. And so a key thing here, though, is that like the brain tends to really get bored, at least according to the adaptation hypothesis. The brain really gets bored when it's hearing something over and over again that has the same acoustic properties. And so by acoustic, I mean, if you hear a pitch with the same pitch, with the same frequency over and over again, that the frequency or the pitch, the specific tone, ba, 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 it's like the same thing over and over and over again. That is the acoustic property. The pitch or the frequency is the acoustic property. And a key thing about adaptation is that adaptation is more likely to occur for sensory features. When you're looking at primary auditory areas, which we haven't talked too much about these regions of the brain in this course, but those, by the way, are in superior temporal lobe. Superior temporal lobe, primary auditory cortex, processes a lot of these low-level acoustic properties, such as the pitch that you're listening to. And the idea is that adaptation only occurs primarily for these low-level sensory, that is, acoustic features. And so therefore, the mismatch negativity, which is almost always studied by, the standard has one set of acoustic features, and the deviant will be somewhat different acoustically. You know, ba and da are common stimuli used in the mismatch negativity studies, and they're acoustically different. Your brain can easily tell the difference between ba and da. And the way you're doing that is by, you can hear the acoustic differences between those things. You can categorize one as starting with a B, and you can categorize the other as starting with a D, ba and da. And so the adaptation hypothesis basically says that for acoustic stimuli in particular, that is sensory stimuli or low-level features, that the brain adapts. The responses become less and less and less to those low-level sensory acoustic features of a stimulus. But then when something new occurs, when something deviant occurs, the brain responds as though it's a new and interesting thing. Basically, the more something occurs, the less the brain's response. When something is rare, the brain is more interested in it, and that's what the mismatch negativity, at least according to the adaptation hypothesis, suggests is going on. The brain responds more to the deviant because it's new, it's kind of unusual. The brain is more interested in new or unusual things, and so there's a bigger negativity in response to the deviant. It's more interesting to the brain. The standard is old, or the standard's not interesting, so the brain doesn't respond to it much. Key feature, though, once again, the adaptation hypothesis says that this adaptation should only occur for basic low-level sensory features. There is some evidence in this article, Mismatch Negativity Review 1, which goes against the adaptation hypothesis. And I think I asked a quiz question about this as well, because it's one of the primary points of this paper, is that there are studies showing that the mismatch negativity can be elicited by a violation of an abstract rule. And so I imagine a lot of people here, what is a violation of an abstract rule? You need an example to really understand what is an abstract rule? What does that even mean? And so here's a good example, and I've just quoted it because it's, once again, they do a really good job of they give an example. They say mismatch negativity can be elicited by a violation of an abstract rule. These are not, when the low-level feature changes, that is not an abstract rule. It's not a rule at all. When low-level feature changes, you're relying on just differences in the low-level acoustic features. When an abstract rule changes, you're not relying solely on those low-level acoustic features. So, for example, if there's, it has been found that the mismatch negativity is elicited by an occasional ascending tone or tone repetition in a sequence of regularly descending tone pairs. So the tones tend to go down in a sequence. Their tones are going down, and then you present a tone which is going up. Then the rule is downness. That is kind of the abstract rule. And then, but please notice when it's going down, you have a dun 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 dun. That's going down. But please note, you're not going to be having sensory adaptation because you're not having the same pitch repeated over and over and over again. So dun 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 dun. That's going down, but you're not repeating anything. The only thing that's there is kind of the rule going down. And then all of a sudden, if there's a tone repetition, so it's going dun 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 dun, and you repeat the same tone, that violates the rule. Or if you go dun 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 dun, and then you go up, that violates the rules as well. So the rule is down, and there's two ways, at least they talk about in this study, you can violate it. One is just by repeating a sound twice. So it's, you're going down, but then you repeat a sound twice, so it stops going down. Or you're going down, and then you can go up. You can kind of go against the pattern. It was going down, but then you go up. Those are violations of kind of the down rule. And you can get in a mismatch negativity in response to those pitches, which kind of violate the pattern. And once again, though, these patterns, the standard is not presenting the same thing over and over and over again. So you're not getting that low level adaptation. Your brain is kind of internalizing the rule. And so this is the evidence. You're not just using low level sensory features. Your brain is kind of internalizing the rule. And so this is the evidence that was against the adaptation hypothesis, because it's not a single pitch which is being repeated. It is an abstract rule. And the brain can respond with a mismatch negativity even when it's a violation of an abstract rule. Let's see, how are we doing on time? All right, yeah, I do want to talk about the early right and anterior negativity, and then we'll call it a day. So this is the last one for today. So this one goes along pretty nicely with the mismatch negativity. In fact, one theory, it's got a different name, and it's this ERAN is applied solely to the music perception domain. One theory basically says that this is the exact same thing as the mismatch negativity. But other theories say, oh, the ERAN, the early right anterior negativity, is a completely different thing. But it has a lot in common with the mismatch negativity. And I guess I didn't list all the things that it has in common, but one thing that it has in common, which is very obvious, the ERAN, early right anterior negativity, is a negativity. It occurs with the somewhat similar, just like the mismatch negativity, it occurs with a similar latency to the mismatch negativity. So the latency of the mismatch negativity is like between 100 and 250 milliseconds. The ERAN is described as having a latency between 150 and 250 milliseconds. So it has a similar latency of the mismatch negativity. And another really important similarity between this and the mismatch negativity is they are both elicited by deviant stimuli, things that kind of violate the expectation that was established earlier in the context. There's, one or two important differences about the ERAN compared to mismatch negativity. And so the ERAN has been studied specifically with respect to musical sequences. And so this is a good time to make the connection between something that we talked about way in the beginning of the semester. We did a lab with the probe tone task. This is where we would present a melody, and then there'd be a single pitch at the end of that melody, and you'd have to indicate how well did that single pitch fit with the context that you just heard. And remember, some of the pitches, like the do note, the do, re, mi, fa, sol, la, ti, and then we could play a bunch of different notes here, and then you rated how well they sounded. And the class, very strongly supported the idea that the do note was the note that fit best here. So that would be the expected note here. And then all those notes that were out of key, when those notes played here, you guys all rated them, not all, but most of you guys, like 90, 95%, rated those notes as sounding very out of place. So this is something which is measuring your kind of pitch expectations. There's a context of notes, and then we're measuring your behavioral response based on the probe tone rating. How well does it fit on a scale of 1 to 7? And we showed that you guys definitely could tell the difference between those expected and unexpected notes based on your probe tone ratings. We also talked about harmonic priming task. And this is where we read about an article early in the semester, a sequence of chords. And then at the end of the sequence, there's like a target chord that you have to make a response to. And usually the target chord, so like the sequence of chords would be played on piano. And then at the end of the sequence, the target chord would be played on one of two different instruments, like a guitar or a harpsichord. And your job as the participant is to indicate as quickly as possible what was the name of the instrument that produced the target chord. Was it a guitar or was it a harpsichord? And then the important variable in the harmonic priming task is, was that chord, the target chord, was it the tonic note, the tonic chord, the one that was supposedly very expected, or was it a chord from outside of the key, one which would be relatively unexpected? And you're making that judgment. One instrument is playing the target chord, and what we read about, is that participants' reaction times were significantly faster when that target chord was expected, given the context, and they were relatively slower at responding to that target chord when that target chord was relatively unexpected. The ERAN, the earlier right anterior negativity, is basically a brain response which mirrors kind of these behavioral findings quite well. In general, people process expected musical events one way, and they process the unexpected musical events a different way. The ERAN is nice because it responds differently to expected versus unexpected musical events. So the brain can differentiate between unexpected and expected things, just like behavioral responses do. It seems like there's probably some type of a connection here between the behavior and the brain. There's definitely some evidence that that's true. So the ERAN, I've kind of already talked about it, is a negativity, has this latency right here. And the negativity, just like the mismatch negativity, it's bigger in response to the deviance. It's bigger, the negativity is, in response to the things that are unexpected given the context that just occurred. So you read this ERAN 1 article, and for that article, the out of key chord was called a Neapolitan chord. So I'm sure you don't, it's a chord I've never heard about, and I'm a musician, so I'm sure you guys don't know exactly, probably most of you guys don't know music theoretically what this is. You don't need to either. Just need to know this is the unexpected chord. Out of key or Neapolitan is the unexpected chord. It elicits a large negativity during this window of time, whereas the in key, that is the tonic chord, it basically elicits no negativity, or at least a much smaller one relative to the Neapolitan or the out of key chord. So here are this is also pertains to some of the questions that I asked regarding this quiz. So there was, in this study, there was a significant interaction of chord and hemisphere. So this is a two-way interaction. And this is this is like quoting directly from the article, indicated a right hemisphere predominance of the early right anterior negativity. But this is a typical two-way interaction, though it might be a little bit hard to kind of get your brain around it because we're dealing with neural measures here, and neural measures are usually a little bit more complicated than behavioral measures. So I wanted to break this down a little bit so you can understand what this two-way interaction actually means. So the ERAN is, by definition, the difference in amplitude between these two conditions right here. And it's shown this is an electrode, this is a different electrode, and this is a different electrode. F7 is a frontal electrode in the left hemisphere. FZ is a frontal electrode in the middle of the brain, the center. And F6 is a frontal electrode in the right hemisphere of the brain. For the purpose of this slide or this interaction, you can actually completely ignore this. This middle FZ electrode, you do not need to pay attention to that at all. All we care about are the two electrodes, the left frontal and the right frontal. And what we care about is, first, let's look at the ERAN. Remember I said it has a latency of around 100, 200 milliseconds or so. And you can see it right here. So that is the ERAN right here, is the difference between these two waves right here. And this is a really key thing. The ERAN is not just this wave right here. It is the difference between these two waves. If you counted the peak here to the peak here, that full amplitude difference, that technically is the ERAN. And let's look at what these two lines mean. The in key one, that's the dark lines right here. That's the brain's response to the tonic, the chord that was expected. The dotted line is the Neapolitan chord, the out of key chord, the one that is unexpected. And I need to mention, this is a graph where negative is up, positive is down. Negative is up, positive is down. The dotted line right here, that's the out of key chord, the Neapolitan. And you can see there's a big negativity right there. And then for the in key chord, there's arguably actually a slight positivity for the in key chord. And so the ERAN is basically the relative difference in amplitude between these two conditions, the out of key condition, Neapolitan, and the in key condition. The ERAN is the amplitude difference between those two. And if you look, there's a big difference right here in the right hemisphere. If you look in the left hemisphere, you can see that there's still a difference between these two, right? There's still a significant, amplitude difference. But if you look, the difference here is relatively smaller than the difference here. And that is exactly what this interaction is showing us. The difference in amplitude between the in key and out of key chord was bigger. This difference was bigger in the right hemisphere than it is in the left hemisphere. So this is just another two-way interaction, but I've always felt that brain interactions are a little bit more complex. We've been talking about interactions all semester. And one of the things I definitely want you to understand by the time we're done in this course is how to read a graph like this. And if I say that there's a significant chord by hemisphere interaction, it'd be great if you'd be able to explain what in the graph indicates that there is. What do you see in this graph which indicates that, yeah, there is this two-way interaction? The answer, the difference in amplitude between these two conditions is bigger in the right hemisphere than the difference between these two conditions is in the left hemisphere. Actually, I don't want to completely finish. I feel like this has been a lot of dense stuff, so there's not much left on the ERAN. We'll start here next time. And then we've got some more ERPs to talk about. We'll probably be talking about ERPs the rest of the week. I will be posting some quizzes for you today. They'll be due at the end of the week. Now I believe I have a couple meetings. I think I have a meeting with the group right now, and then one or two students after that meeting. So I'll turn off recording now.